Hello everyone, I am Dr. Ginny Toma and I'm here to give you a brief idea about bioinformatics. So the title for my today's talk, as you can see, is Introduction to Bioinformatics. Bioinformatics, as you can see, is made up of two words, bio and informatics. This implies we can assume that we are trying to study biology with the help of information technology. That includes the study of the various concepts of biology with the help of computers, information technology, mathematics, and statistics. Let's have an idea of what does bioinformatics include. Bioinformatics is an interdisciplinary field which came into existence to bridge the gap between the computer scientist and biologist. There were few terminologies which the computer scientists weren't able to make out the difference. If there is a change in one base pair, then how can it lead to major changes in the biological data set? Likewise, biologists weren't able to convey what exactly they want, what kind of output they require from the computer scientist. Thus, in order to bridge that gap, bioinformatics was introduced. So, it is an interdisciplinary field which includes the study of different biological aspects using computational biology, or we can say computational resources. As we all know that in order to work in any language, we need to know the basic alphabets, words of that particular language. In the same manner, when we talk about bioinformatics, in order to work in the field of bioinformatics, in order to utilize the various resources provided by bioinformatics, we need to know what type of data is actually required in case if you want to explore the various facets of bioinformatics. So the first thing which a user, a student, a researcher need to know is what is the type of data which is required in order to work in bioinformatics or in order to work on the different tools in bioinformatics. So if we want to categorize about the type of data, we can broadly say there are two categories. One is sequence, another is structure. When we say sequence data, we are talking about either nucleotide sequence data or protein sequence data. When we talk about structure data, prominently we are talking about the protein 3D structure data. One thing which should strike you as from where we can get this kind of data set. So let me tell you that when we talk about bioinformatics, we are prominently talking about the various biological databases and the numerous tools which could be free or paid version, and then analyze our biological data with the help of those tools. So from where we can get these biological data set? We can get this biological data set from the biological databases. In order to work in bioinformatics, three main organizations are able to give a extensive idea about what all different categories of subdata is required in order to perform any bioinformatic based analysis. That includes NCBI, which is National Center for Biotechnology Information. Another main organization is EMBL, European Molecular Biology Laboratory. Its European Bioinformatics Institute sister concern is dealing with bioinformatics related data. And the third main organization, which is working in joint collaboration with these two, is DNA Data Bank of Japan. All these three together make International Nucleotide Sequence Database Consortium. So their purpose is to reduce the redundancy, to have similar kind of data. Now, when I'm saying similar kind of data, I meant that if I am working on X organism and I have the sequence data after performing sequencing and other techniques, and I want to deposit it, submit it to NCBI, which I find more comfortable, and I've submitted the information. And somebody at some other place is also working on X organism, and he or she has already sequenced the data set the same manner in which the entire team, were able to do. And they have submitted the data in DDBJ. So now there will be discrepancy, there will be repetition. So all those people who will take our data set and then start working on it, and all those people who will take their data set and start working on them, will at one point of time will feel that the two data sets are similar, and thus many people will get affected by it. So in order to reduce such cases, INSD was established, and these three organizations are working in joint collaboration. So that's a way we have covered the second point, which is source of data. Now, as we have already talked about that in order to work in a language, you need alphabets. We already know the alphabets of bioinformatics, the sequence and structure data. From where we can get it? The next step is sequence analysis. Sequence analysis starts with sequence alignment. When we are aligning two or more than two sequences in such a manner so that we can have maximum similarity or dissimilarity between our data set. The data which we want to study is usually referred as query data set. So here I've just given you an example that when we try to uh, study sequence alignment on the basis of the number of sequences involved, we say we are working on either pairwise sequence alignment or multiple sequence alignment. Pairwise sequence alignment, as the name indicates, includes two sequences. Multiple sequence alignment, on the other hand, again, as the name indicates, is making use of more than two sequences. Sequence alignment has been further categorized on the basis of length of query coverage as local sequence alignment and global sequence alignment. So when we say local sequence alignment, what I meant was if I want to study my query data and my query data is from say 1 to 250 base pair. So if I want that at least a part of the sequence should be able to match with some sequence so that I can get minimum information, at least I should get some information about my data set, then I would prefer to go for local sequence alignment. On the other hand, if I would want the entire sequence length, that is from first base pair to 250 base pair to 50th base pair, then we say that we are working on global sequence alignment. So if we want to summarize what bioinformatics include, it includes the study of the application part of research, which includes biostatistics, which includes biomedical data science, and definitely the main part, data mining. And all this thing cannot be covered if we do not have an idea of what are the main concepts related to biology, medicine, health sciences, social sciences, and the genomics or the genetics or the proteomics or the transcriptomes associated with our study. So here we can now easily say that bioinformatics gives us an overview, including both the services of computer science, mathematics and statistics, and biology. If we want to summarize again what could be the basic applications of bioinformatics, then we can easily see here that if you are able to study the genes, genomics part, or the proteins, proteomics part of plants and animals, there we can apply bioinformatics. In order to study the microbial genome for bioremediation and other kind of purposes, we can study using bioinformatics too. We can apply bioinformatics in order to study agriculture-based studies in order to enhance by choosing specific genes, by identifying which genes are responsible for it using bioinformatics tool. we can perform the entire system study. So when I'm saying the entire system, I'm talking about the entire system of an organism. It does not just include one part of it. It includes all the organ system, the networks, the protein interactions, the gene-protein interactions, and so on. There is another point which is phylogenetic analysis. So as you can see here, phylogenetic analysis is actually the study of ancestral relationship through which we are able to identify what is the normal progeny rate of evolution which is being followed. Now, if we want to talk about in detail about the various advanced bioinformatic application, it could be done with the help of microbial genomics, comparative proteomics are the basic part of performing any bioinformatic based analysis. For this, you can use clinical microbiology and see what kind of stress is involved, and then obviously it involves the study of molecular microbiology. You can identify certain motives, you can identify transcription factors, and all these are done with the help of machine learning based softwares. So here we can say that when we talk about bioinformatics from basic till advanced, it is very huge subject. You so it'll take some time in order to understand the basic concept. Once you start working on it, things will improve drastically. So this was my first video demo for the introduction of bioinformatics. Still, if you feel that there is something which is missing and you people are not able to understand clearly, then please write in the comment section. Thank you so much for listening to me.